are any obvious questions from the floor? Okay, seemingly not. I'm sure I've got some. I was very intrigued with the small flowers. I really did. Could you add anything more about these, the structures, and whether they could be useful for other sort of drug delivery applications?
I haven't really done the experiment regarding drug delivery or something like that, but I report the new method in synthesizing high porous small flowers, and recently I find out the clue to control the size, even the size of the small flowers, which was not reported yet, and we can even generate real nanoflowers to microflowers. So I think I like to, if you're interested, and then I'm not a drug delivery guy, but I can collaborate, and we can develop or add more values to the small flowers that are synthesized.
I think there's a lot of value in essentially self-assembled systems really for drug delivery, and I think I think there's some quite clever systems out there, and I think you could still do a lot more really, and get around this problem of odd stoichiometric mixtures and so on, which are really problematical. Really.
And I also like to mention that I can have a covalent organic framework as well as ionic organic framework. If I use the latter, then I can dissolve the small flowers in the aqueous media when I want to have it dissolved. So if I can imagine that if it can carry some kind of drug in the form of nanoflowers, then we can, after the mission, it can naturally dissolve away, something like that. But that's my dream, so we can have some conversation later.
Any other questions? I can keep going for hours. I'd rather see some from the floor, though. Yeah, there's half a question. It's coming.
Hi, Matthias Roman, IBM Research here in Zurich. Professor Wenzel, one question regarding the modeling of the herd channel that you've been doing. I know your colleague Olaf Dössel is doing hard modeling where the herd channel is playing an important role as well. But his work sits on top of what you've been doing in a way, right? You've done more like molecular dynamic simulations. Yes. And have you thought about combining the MD simulations with the physiological simulations that he's doing?
I mean, at the moment we don't. This is working more or less. So far we haven't, right? We are looking into this, right? In the academic world, it's always the question to get the funding, right? So this is one of these projects which keeps rejected, with encouraging, comments how to do it better next time. So hopefully in a year or two, in many other aspects, we're going sort of into this multi-scale dimension because you can, never go into this 3D cell size or vesicle size type of modeling on this molecular basis. It's also pointless, right? We've been working a lot on sort of implicit models, at least for membranes and stuff like that, that help us to stay still molecular, right? But focus on the parts of the system which are actually functioning. To make the challenge sort of to the sort of hydrodynamic or finite element level is here very difficult. Yes.
Do you have any thoughts on modeling? I mean, I was in charge of modeling in my company for many years, and I saw the problems and the advantages of it, but are you capable yet of doing modeling self-assembled systems, really? It's slightly outside the scope of your talk, really.
We've done it for DNA-based materials, but they were sort of very experimental, right? Not in any living environment, right? It's a challenge, right? Because I think what we always have to balance is specificity, right? Which typically arises on the molecular scale, right? And the problem of going coarse graining that is that you typically lack the force fields, or the interaction mechanisms, right? And when you stay on the molecular scale, then the problem is the time scale, right? Because the things that we can simulate are by now quite large, but the times that we can simulate are quite short. And so therefore, the problem is really to try to find methods that extend actually reach the timescale on which assembly takes place, right? You can cheat in certain applications, right, by accelerating the simulations, but this only works if you know where you're going, right? So if you have like protein folding and it's folded or unfolded, or receptor ligand binding where you have a very specific process where you kind of know reaction coordinate, then it's good. But if you have just assembly and you want to predict what it assembles to, right, then typically all of these things don't work.
That's the same sort of answers I got really
Yeah,
at the
yeah,
time. People either say, we don't understand the force field, or we need a lot more money spent on computers. There we go.
Yeah.
Can I ask one question about the maleimide? What your thoughts on that and how you might replace that? Because I don't know the solution of that either.
Yeah, absolutely. I think, obviously moving on to amide bond coupling is the next sort of, more robust approach as such. However, I think, eventually, in the first instance, we're trying to sort of do away with post-coupling chemistry, which is a lot more difficult, to sort of, in a way, control for. And, our methodology avoids sort of like post-purification, post-nanoparticle purification, and all that kind of thing. So, I would say, moving towards maybe trying different linkages in a sense.
I mean, in its favour, maleimide can be used for industrial processes, and it's extraordinarily fast in its reactivity. And whatever you do to the system, it seems to be less reactive or have other problems, really. But the downside is it does reduce your shelf life unless you can keep the pH very acidic, really. Even then, it goes off.
Absolutely, but I think you make a great point about the linkages that you use. I mean, I've done folate lipid conjugates in the past, and we've had these liposomes tested by the NCI in the US, and they've actually found toxicity relevant to the linker itself between the ligand and the lipid. So these are all points to take into consideration and test out.
I think there's a scope for new chemistry there, but it is not an insignificant challenge, that's for sure, really. So.
My question is to Dr. Kamali. I would like to know that you have been doing a chronic treatment when you are treating atherosclerosis. What kind of route of administration you are using for that?
Absolutely. So we're going advanced atherosclerosis because that sort of models the more vulnerable stage of the plaque in patients. And we use tail vein injections.
But tail vein injection every day, is it possible? Means I have been working with in vivo models in my past, and I see tail vein injections are not really, they don't work every day for the chronic treatment. We had to go shift from trained vein injections to the IP treatment, interperitoneal. So,
so you mean it causes aggregation with respect to the nanoparticle?
Right.
Yeah, I mean, obviously there are inherent factors like that in any route of injection with nanoparticles. Whether IP is the better route or IV, I think we've also done IP experiments, and this sort of really changes the pharmacokinetics of the drug payload. And in our case, we want to sort of mirror what can be most easily translated to the clinic, and that is systemic injection. However, we are moving more towards oral delivery and developing oral platforms, and so really that's kind of like the next big thing.
One more question with that. Did you study with the retention or the clearance of the particles which you're treating? I mean, after treating, we just calculate how much we are administering, but how much is actually causing the effect in the atherosclerotic area? Means in the intraperitoneal route or IV route, some of the particles are retained and goes to other tissues of the body, and there is a particular amount which is very difficult to calculate. Means I face this problem in my in vivo studies, so I just want to know whether you did that or not.
Sure. I mean, based on the fluorescence signal of the nanoparticles, and this is a fluorophore conjugated to a protein that's inside the blend of the polymeric composition, we actually observe up to 70% of the dose in the aortas compared to the spleen and the liver. So, based on fluorescence, we have to obviously validate this further and have a more robust kind of, maybe look at the peptide itself fluorescently and so on. But yeah, that's a good point.
Yeah, because the percentage is quite not clear how much it really works there and doesn't work there.
Absolutely.
Thank you very much for your answer.
You're welcome. Thank you.
To ask Stefan about his USP in terms of targeting. How does the carbohydrate targeting compare with, say, an antibody targeting? So really, what is your advantage, really, over the existing products, really?
Well, antibodies need to be produced in cell culture, and sugars, it depends where they come from, but the ones we use are synthetics and quite affordable, I would say, in a kilogram scale. Glucose is very cheap, and there's others that are like 1,000 francs for a kilo, but still, I think you can get even a meal from that.
Antibodies are very cheap, and I think I said earlier, really, it's surprising because fermentation, you don't need chemists.
Yeah,
and it's scalable
process. The thing is, we're using synthetic biology. So there's no cell culture, no bacteria ever involved in the whole production process, which facilitates a lot the manufacturing for us. And I think also the size of the molecule itself and its immunogenicity should be at some level respected, because sugars, they are that much present, you can barely get an autoimmune reaction to it. And this allows for repeated administrations in a very broad manner.
It reminds me that I was in Spain, I think a year or two ago, and somebody said they were starting up a aptamer project which could be cheaper because antibodies were so expensive, but it's in fact the other way around. DNA is much more expensive than antibodies. They cost about, at scale, something like $50 a gram. So it's really relatively cheap. Anyway, I think that concludes this afternoon's session. If there are no more questions, and we're bang on time for tea. So could I thank the speakers very much indeed for a very diverse range of talks. Thank you very much.